Hi, I'm Fyodor Urnov. I'm a professor at UC Berkeley and a director at the Innovative Genomics Institute. And more importantly, I am one of the co-organizers of the Keystone Symposium on the Precision Genome Engineering, which will be happening early in March in wonderful, beautiful Green Ireland. Make CRISPR the standard of medical care. CRISPR has given us curative outcomes for sickle cell disease, amazing data in late-stage clinical trials for degenerative disease. Now, how do we benefit the entire planet? 300 million people, 5,000 genetic diseases. So the vision for the symposium was really to bring sort of an end-to-end constellation of scientists, physicians, regulators from early stages of discovering what to edit to taking it into the clinic so that we can learn from each other. I don't want to impugn all of the other meetings, they're great. I think ours is distinctive in that we start at very foundational science, like how to use CRISPR to discover a gene editing target to treat a disease, and then go very, very translational, all the way to how you design a clinical trial to treat somebody with CRISPR. Every step in the journey from a patient with a genetic disease to a CRISPR cure has had something remarkably innovative happen. And so all of that's, I'm very happy to say, represented in the program, from the extraordinary use, for example, of organoids to understand disease genetics, to pretty phenomenal ways to do high-throughput screening, to new fundamental tools to evoke curative outcomes in genetic diseases, things like genome editing and others. And then going to sophisticated animal non-clinical models for how you actually build a medicine for an ex vivo or in vivo disease indication, and all the way how do you measure readouts in a clinical trial. All of that has been innovated. In other words, the 2025 version is certainly not the same as the 2023 version. And it's such a thrill to be able to combine all of this innovation under the roof of one symposium. So we will emerge from there 3 days later, kind of riding this wave of exhilaration of how much new stuff there is in our field that we can put to use. I am very eager to hear people nominate new diseases for CRISPR cures and learn from others at the meeting to say, oh, I work on this disease of the immune system. I hear you have a new tool. Can we collaborate? Or, oh, you have a new delivery modality that makes this organ suddenly deliverable to. Well, I have patients with that disease. So I would love to be a fly on the wall at those conversations. The single most impactful meeting of my scientific life, this is true, was when I was a third-year graduate student and it was a Keystone Symposium. I was a wet behind the ears kid. I had no idea in retrospect. And I just remember immersing myself for several days, bumping shoulders with the scientific celebrities. And I still have my notebook, it's this thick, with notes I took. I cannot recommend strongly enough for early stage trainees to come. Bring a poster. You have until February 10. It does not have to be a cover of a journal. Show us some data. You'll see an incredibly friendly audience eager to learn what you've discovered and perhaps offer some advice. I hope the biggest impact from this will be the emergence of collaborative solutions through which we can both advance quote-unquote academic innovation in this space, but also give the for-profit sector tools to connect with, to potentially accelerate their own pipelines. Nobody doubts that in 50 years, CRISPR will be everywhere. Question is, how soon is that future going to be here? I really think that we will kind of inflect the curve of CRISPR adoption in biomedicine in a meeting such as this. If you are interested in seeing how gene editing will impact public health, come to the meeting. If you're a fundamental scientist interested in building new ways to measure specificity or sensitivity or new delivery tools or new editing modalities, come to the meeting. If you build sophisticated models to model disease in animals or in a dish, come to the meeting. If you work at a biotech company hopeful to enter the gene editing space, or if you're a physician who has patients who could benefit from CRISPR, or if you're a graduate student who's trying to decide, should I go into CRISPR as a tool? Come to the meeting. We live in complicated and uncertain times. All sorts of things are happening in the United States, across the world. And you look into the future and you don't know what's going to happen. Genetic diseases will be with humanity for as long as humanity exists. CRISPR gene editing is here to stay. And I think If you think about Springsteen's famous line, at the end of every hard working day, people find some reason to believe. I think the fact that CRISPR plus genetic diseases will equal curative outcomes is our reason to believe. And I'm really hopeful that coming to this meeting will give you a very realistic sense of the extent to which that is true and help us all persevere through the complex and uncertain times we live in.